Hey guys, it's Bernice, and in this video I'm going to talk about DNA replication. To begin, what is DNA? DNA is deoxyribonucleic acid, and it is genetic material found in organisms. If you zoom into DNA, you will see nucleic acid monomers called nucleotides, and it consists of three components: nitrogenous bases, deoxyribose sugar, and phosphate. There are four types of nitrogenous bases: adenine, guanine, thymine, cytosine. Adenine pairs with thymine, and cytosine pairs with guanine. Now that we know what DNA is, let's move on to DNA replication. Now, DNA replication occurs during the S phase, and it is semi-conservative. That means that each DNA molecule is comprised of one old strand and a new strand, and the parent strand will be a template for the new strand being formed. There are three phases of DNA replication. First is initiation, second is elongation, third is termination. Initiation. The DNA is unwound at the origin of replication, which is a specific sequence of DNA that is the starting point. This is possible because of an enzyme called helicase that breaks the H bonds or hydrogen bonds between the base pairs. Single-stranded binding proteins then come to help stabilize the unwound single strands by preventing them from annealing. Unwinding the strands essentially forms a Y-shaped structure called a replication fork. Because of the unwinding of DNA strands, it may cause tension, so topoisomerase relieve the tension in DNA strands by cutting one or two strands near the replication fork. And this allows the strand to untangle, and then it rejoins the cut strands. Helicase can separate DNA in both directions of the origin, and it can do that in multiple locations. When two replication forks proceed in opposite directions, it makes a replication bubble. Next is the elongation phase. DNA has a 3' end and a 5' end. The 3' end essentially has an OH as its last group, while the 5' end has a PO4 as its last group. Remember that these DNA strands are anti-parallel. That means that one side is 5' to 3', while the other side is 3' to 5'. Also keep in mind that DNA is always synthesized from 5' to 3'. So let's begin synthesizing. To begin, the RNA primer must first anneal to the template strand and then is attached. And this happens by this enzyme called primase. These RNA primers are essential because DNA polymerase can only extend a nucleotide chain, but it cannot start one. So therefore, in order to synthesize new DNA strand, it needs to extend from a RNA primer, and of course in the 5' to 3' direction. Eventually we'll have to take out the RNA primers, so that's when we use DNA polymerase 1, and it removes the old RNA bases and replaces them with DNA, which leaves a tiny gap. That's why we need DNA ligase to join these two fragments together by forming a phosphodiester bond. The nucleotides are always added to the 3' ends by DNA polymerase 3. The strand with the free 3' OH end is called the leading strand, and is essentially a DNA strand as assembled continuously in the 5' to 3' direction towards the replication fork, whereas there's a strand with no free 3' OH end, and that is called a lagging strand, and with this DNA strand is assembled discontinuously in 5' to 3' direction away from the replication fork, and this essentially causes small fragments. So let's zoom in into the lagging strand. Assembling DNA strands on the lagging strand we need to add RNA primers, and this is because there are no 3' ends on the liking strand. When we do this, the DNA polymerase III adds in nucleotides until it hits another RNA primer. From there, DNA polymerase I removes the RNA bases and replaces them with DNA. This does leave gaps, which make Okazaki fragments. The DNA ligase links the Okazaki fragments by forming phosphodiester bonds. Last but not least, we have the termination phase. So as soon as these strands are complete, they rewind into a double helix structure, and the two new DNA molecules separate from one another. However, sometimes in DNA replication, there may be mistakes that occur, and a wrong nucleotide may be placed. Therefore, we have two mechanisms to counteract this problem. The first one is by using DNA polymerase III, and essentially it can reverse and replace the nucleotides during replication. However, what happens if we need to fix a nucleotide afterwards? Don't worry, because we have DNA polymerase II, and it can replace the nucleotides after and fix it.